welcome back to the NPTEL lecture series on animal physiology. So, we are in section 12 with the lecture 3. This is basically in the gastrointestinal physiology. So, we talked about in the first lecture we talked about the overall architecture of the digestive system. We talked about how the nervous innervation of sympathetic and parasympathetic systems are kind of controlling the whole system, the whole architecture. And then on in the second lecture we talked about the first part where the first intake of food takes place, that is in the mouth, in the saliva, the function of the saliva, how saliva secretion is being regulated, and how the first phase of the food is being broken down into pieces before through bolus movement in the form of peristaltic movement it moves to the stomach. The stomach is the, I should say, the first after the mouth, it is the first station. Through the esophagus it reaches the stomach. Stomach is the first station where the food is exposed to one of the most acidic environment in its whole tract, all the way from mouth to excretion from anus. This particular structure is the most acidic, where the pH almost hits upon 1 and even possible less, and that is because of hydrochloric acid, which is being produced by their different cells, their parietal cells, Ceph shells and everything. So, the way we are dividing this particular class is, we will talk about the anatomical feature of stomach. From there we will move on to the different cellular structure and how they regulate the movement of H plus and Cl minus ions, which leads to the formation of hydrochloric acid, and then we will talk about the different phases which regulate the whole digestion process. Okay, so, anatomically speaking, so if you look at the structure, so let us get back to the section 12. Section 12, lecture 3. Stomach. Okay. So, the overall structure, if you look at the stomach, the way it looks like, it is a structure, it physically looks something like this. Okay, and of course not this. One second, let me just redraw it. Something like where the food moves through the whole path. No, wait, this is the wrong way to put it. Okay, let me redraw it. Okay, something like this. Okay, so the food is coming. I am showing you the arrow. This is how the food is entering, and this is where the food moves to the small intestine. And whatsoever happen, everything happens in this zone. Okay. So, if you look at the very basic structure of it, it is divided into 4 different parts. The parts are named as cardia, fundus, the body, and the pylorus. And pylorus. These are the 4 anatomical feature of stomach, and each one of them have their own function. And what I will do, and there is something called pyloric sphincter, which is present somewhere out here, which ensures this pyloric sphincter ensures that when the food should be sent to the intestine till, so it is kind of a control mechanism by which food is being ensured that all the gurgitation, regurgitation, all the chemical reactions takes place in such a way that the food is now ready to be transported to the small intestine for further absorption and all other metabolism related events. Okay. So, now coming back to the structure, the anatomical feature of the structure. So, let me redraw this so that I can label all it for you people. So, okay, fine. So, this is the entry port of the food. So, this is esophagus. So, then the food is entering, this is the fundus region, and this whole vessel, if I have to take a cross section of this, something like this, if you see if I take the upper view of it, you will see lot of blood vessels all over the place, likewise. Okay. It is completely innervated with blood vessels, likewise. So, this is just the kind of a cross view what I am trying to draw for you people. If you get an upper view, then this zone is called cardia, and here you have a bunch of longitudinal muscle layer. There are two kinds of muscle arrangement here, longitudinal, and these are all smooth muscles, longitudinal muscle layer, and second one is series of circular muscle layer, and these longitudinal and circular muscle layer helps in the movement of the stomach, circular muscular layer or muscle layer. Okay. And then you have, this is the fundus, this is cardia, and here is the body part, and this surface is called rugae surface, and this zone is called pyloric antrum, and here is the pyloric sphincter, and this part is called pyloric canal. So, this is the overall geometry of the stomach. So, all these organs, most of these digestive organs or any of the secretory organs needs a site or needs a geometry or anatomy by which it can secrete. So, think of it, you have, so when you have to, if you think of a pipe, okay, what happens? A pipe, it's a hose, and it started dumping out the fluid. So, what kind of pipe like structure this stomach has? So, there we are entering, after giving you this whole overall anatomical idea of it, I will move on to the cellular structure, how the cellular structure of stomach looks like. Okay. Let us move on to the cellular structure and how the histology and the cell and where all these different cells are. So, basically what we will be doing, we will take up a cross section of this and we will move on to the cellular histological feature. Okay. So, the cellular structure, the way it looks like, if you go through the cross section of the cellular structure, it looks something like this. It is a bit of a complex drawing, but bear with me, eventually it will look very straightforward. So, something like this. This is how the structure looks like, and even if you make a cross section, it will look like this only. Okay. So, and these are lined by different cells like this. Okay. And I will give the different nomenclature. So, imagine it, if we reverse it, it looks more like a pipe. Okay. So, I am just drawing one of them, and I will leave the rest for you to, for your imagination to take over. Okay. These are the different cells which are forming the structure. Okay. These are the nucleus I am putting. Okay. So, these are called these structures are called, this gap what you see is called the gastric pit. Fine. And these cells are called mucous cell lining, mucous epithelium. And within the mucous epithelium, you have some series of cells, and underneath it, and I will come in depth after, I will just give you the overall. Underneath this, you have all the blood vessels which are moving through all this. Likewise, these are the blood vessels, you see. And along with the lymphatic vessels which are running along. Now, if I take a complete cross section of this, something like this, and redraw it, how that will look like. So, where I will be talking about the different cell types which are involved in it. Okay. So, that will look like more like this. Again, redraw that one small fragment, that will help you to realize it better. Okay. I just picked up one of those gastric pits. Now, let us put all the cells which are present in the gastric pit. I am just doing it from the reverse side, drawing the cells. That makes life a little bit easier. So, these are the cellular lining which are forming the gastric pits, and underneath the gastric pits you have another series of cells which are forming the supporting cell to the gastric pits. Okay, and so these are the nucleus. Okay. So, in this, so I told you that these are the mucous cells, and this zone what you see here is called the neck of the pit, and within the neck of the pit, underneath that you have some cells which I am just now putting in red. These are called parietal cells. Parietal cells, and underneath that I am putting them in green, and the series of cells which are called Sheff cells, and you have series of smooth muscles around it, and very underneath out here you have certain cells which are called G cells. So, each one of these are formed of, each one of those pits what I have drawn formed out of Sheff cells, parietal cells, and the G cells. So, we started with the overall architecture. We talked about the position of the pylorus, fundus, body, and the connecting tube. Okay. And then we talked about the basic cellular anatomy, and we showed you the location of the gastric pit. So, through these gastric pits what I have drawn, basically through these pits you see the secretion of hydrochloric acid, how that happens. Now, we will move on to the production of hydrochloric acid. None of the cells which are involved in it, they do not produce hydrochloric acid. They produce, let me give you the way they do it. They produce H plus ion and Cl minus ion, and they secrete it, and then they interact and form hydrochloric acid. So, if a cell in its cytoplasm starts producing hydrochloric acid, then that cell is, it is very difficult for that individual cell to survive. So, they do not do it like that, and it is the mucus cell lining which prevents these cells from getting destroyed. Okay. So, now what we will do, we will pick up the individual cell here, and then we will talk about how it is forming the, how it is helping in the formation of the hydrochloric acid. Just going back to the cellular geometry of it. Okay. So, we talked about the parietal cells. So, within the gastric pit, now we are inside the gastric pit. So, within the gastric pit you have two types of secretory cells. So, first one is the parietal cells. These parietal cells are especially common along the proximal portion, or you must have seen that in the picture. So, these are the proximal section where they are fairly common. They are along the proximal portion of the gastric pit, and they secrete intrinsic factors like vitamin B12. Okay. If you remember that, this was helpful in vitamin B12 is helpful in the RBC production. Okay. So, parietal cells, the other function of parietal cells are HCl production. Okay. So, how it does so? So, let us look at the geometry of the parietal cells. So, I am at the cellular level. What I have drawn just before this, now I am talking about the individual cells. So, let us take up this section. Okay. Now, what is happening is that we have talked about this reaction CO2 plus H2 in the presence of carbonic anhydrase, enzyme, it forms H2CO3. H2CO3 immediately dissociates into H plus and HCO3 minus ions. Okay. Then what happens once this gets dissociated? So, this bicarbonate ion which is released here. So, there is these ions, there are 2-3 things happen. This H plus ion through transporter moves inside. So, this side is the lumen side of it, and this side is the interstitial fluid. Okay. So, essentially what is happening is that these two ions are transported independently by a mechanism. Okay. So, hydrogen ion generated inside the parietal cell as the enzyme carbonic anhydrase converts the carbon dioxide and water to carbonic acid, which is H2CO3, what you could see here. Then the carbonic acid promptly dissociate, okay, into H plus and HCO3 minus. Okay. The hydrogen are actively transported into the lumen. So, this hydrogen is actively transported into the lumen area. Okay. And whereas the bicarbonate ion which are produced here is CO3 is transported into the interstitial fluid. So, they follow a two different. So, there is a counter transport mechanism. So, this is a classic case of between these two, this one and this one, there is a counter transport. Okay. So, after this what happens, this H plus which is being moved out there from outside, from the interstitial fluid, the chloride is being transported like this, and this chloride transport and HCO3 ejection is a reversible. So, on one direction what is happening is that the bicarbonate is being released out of the cell and chloride is being taken up the cell. Okay. So, it is a kind of a, so there is a mechanism by which there are two reverse processes of which are happening. So, chloride is getting inside the cell and bicarbonate is sent out of the cell. counter transport. Okay. There was a co-transport, there is a counter transport taking place. Okay. So, after this what happened is that once this chloride is being sent here, then this chloride, so diffusion process moves inside the lumen. So, now you have the source of H plus and Cl minus, and this side what you are seeing, this side is the pit. So, this is where they are throwing out these two, and whereas when this HCO3 is coming on this side, this leads to an event which is called alkaline tide. This makes the interstitial fluid fairly alkaline. There is a sudden influx of the bicarbonate ion. So, what this HCl does? So, the pH goes to 1.5 to 2, and what it essentially does, it kills all the microbes. This is the major job it does. The second thing it does, it denatures the protein and inactivate most of the enzyme of the food. Most of the enzymes of food. Okay. And the third important thing it does is that it helps to break down the plant cell wall and a connective tissue in the meat. So, plant, because we have a lot of vegetarian diet, plant cell wall breakage and connective tissue of meat is being broken down. Okay. And lastly, this acidic environment does a second set of job. Acidic environment leads to the activation of, essentially the activation and function of pepsin, which is basically a protein digestive enzyme secreted by the chef cells which are just underneath it. So, it follows 4 actions. Once it creates an acidic environment by which most of the microbes are being killed. The second thing it does, it denatures all the proteins and the enzymes present in the food. Third thing it does, it get rid of, it breaks down, chops down the plant cell walls and the connective tissue of the non-vegetarian part of the food. And lastly, what it does, it promotes the secretion of pepsin, which is secreted by the chief cells. So, we talked about the parietal cells, underneath you have the chief cells. So, now we will talk about what the chief cells are doing out there. Okay. So, coming to the chief cells. So, what the chief cells are essentially doing is that they are more abundant near the base, as you must remember, like I was when I was drawing the pit like this, they were mostly concentrated out here along the pit, and this is the pit where all the H plus and Cl minus are falling. Okay. So, this is the HCl pit. Okay. So, they are more towards the base of the gastric pit. This is the major location of it, and these cells secretes pepsinogen, and which is an inactive proenzyme, and it has inactive proenzyme, and this pepsinogen is converted, to pepsin presence of acid, and this acid is being supplied by the parietal cells which are present there. Okay. So, essentially this pepsin is functions most effectively at the pH of 1.5 to 2. That is the zone where it acts, its activity is maximum. Okay. So, we talked about the parietal cells, we talked about the chief cells. Still there is one more cell at the bottom of the pit. If you remember, while I was drawing it, I showed you that there are certain cells which are present here, which I call the G cells. Okay. So, if we go back, you will see that next one. Yeah, look at these are the cells. We have not talked about this. So, we have talked about this, we have talked about this. So, this is the one. So, if I from here coming H plus and Cl minus, okay, and these are the ones which are secreting pepsin. Now, let us talk about the third one in the line, which is the okay, which are the G cells. Okay. So, pyloric gland which is secreting the. So, these are basically they secrete a hormone called gastrin, which is produced by the G cells. And what gastrin does? So basically gastrin is produced by the G cells, and G cells are most abundant in the gastric pit of the pyloric antrum, and gastrin stimulates. This is what gastrin does. So, the function of gastrin is that it stimulates couple of things. It stimulates the secretion of the parietal and the chief cells. So, secretion of parietal and chief cells. This is its major function. This is one of the function of it, and it has a second function. Second function is that, so there is along this there is a contractile movement which is essential. So, contractile movement of the gastric wall. This contractile movement is being supported by this gastrin, and this is extremely essential. It's something like this. The food has to be continuously in motion. It's just like you take, how you mix the dough while you are making bread. It's just you have to mix it, you have to mush it up, you have to really push it through. So, it has to go through this whole motion. That motion is being promoted by this hormone called gastrin, which is secreted by those G cells which are at the base of the gastric pit. So, that is the function of it. Okay. So, now we have pretty much enumerated. There is a couple of more small details which so this pyloric gland also contain, there is something called D cells, which I haven't mentioned in the picture. They are very few in numbers, and they release something called somatostatin, which is an hormone that inhibits gastrin release. So, this is kind of this is the inhibitory signal I am drawing gastrin. So, in other words, the D cells which are present in the gastric pit are regulating the secretion of the G cells which are secreting gastrin. So, it is a auto mechanism which regulates when the gastrin has to secrete, because when the gastrin is secreting, when the G cell is secreting gastrin, so that will stimulate the parietal cells and the chief cells, and eventually the secretion of acid by the parietal cell will be enhanced, and the secretion of pepsin by the chief cells will be enhanced. So, somatostatin is the one which comes into play and ensures that the gastrin secretion is being regulated, and thereby preventing the excess secretion of acid. So, overall, if I had to summarize the cellular structure, so we talked about the anatomy, now we are at the cellular structure. So, there are three types of cells: parietal cells, chief cells, G cells and D cells. Parietal cells are involved in hydrochloric acid formation, chief cells are involved in pepsin formation, and your G cells are involved in gastrin formation, and D cells are involved in somatostatin formation, which regulates the gastrin secretion. Okay. So, now after this, after telling you the anatomy, overall anatomy, cellular anatomy, and the structure of the gastric pit, just one more, just kind of for understanding sake. So, if you look at this structure, which is more like this, so this whole thing has bits like this. these are the pits where all the processes in a 3 dimension. Just imagine this picture in a 3 dimension, just a section I have drawn. Okay. So, from here we will move on to the regulation of the gastric activity, and gastric activity is regulated at 3 different stages. There are 3 different stages. So, one of the stage is called the cephalic stage, and it is called the intestinal stage, and in between there is another stage which will be talking about soon. Okay. So, let us get back to the slides about once again about the regulation of gastric activity. Regulation of gastric activity. So, you have I was telling you there are 3 phases by which it is being regulated: cephalic phase, gastric phase, and intestinal phase. So, what is cephalic phase? It is very interesting that all this secretion process, if we talk about the cephalic phase, starts much before the food reaches the stomach. You might wonder how that happens, because whenever you smell a good food or some really nice something is frying, you get a temptation, wow, I should eat that. By that time, the nerve innervation along the stomach leads to the secretion of a whole bunch of chemicals, and that is where the cephalic phase gets initiated. So, the food has not reached there, by the way. You just smell something and you just taste, maybe you do like this, you taste something and already your stomach is getting ready, oh, something is coming, I have to secrete hydrochloric acid, pepsin, and all other things to ensure I can break the stuff. Okay. So, that is where we are talking about the cephalic phase, and that is why in the first class I was telling you, be very careful, this is very well innervated network of neurons and blood vessels. They are continuously helping this system like an autonomous unit to do its function. So, let us get some of the details of the cephalic phase. Okay. So, cephalic phase. Okay. So, cephalic phase starts with, these are the stimulus, a sight, even the sight of the food is smell, taste, or the most important, thought for food initiates. These are the signal which initiates the cephalic phase, and interestingly what is happening in the stomach during that time, these are the reactions which are taking place in the stomach. So, from the central nervous system, I am putting them in red, in maroon. From the central nervous system, these signals are coming through the vagus nerves. Okay. And they are stimulating the mucus cells, chief cells, parietal cells, and the G cells. Okay. And mucus starts secreting, these are the mucus cells, mucus, these chief cells start secreting pepsinogen, and they start secreting HCl, and the G cells, it starts secreting the gastrin. So, this duration is very short, it is a short duration event, and essentially what is happening during this phase, the stomach is priming up itself that some food will be arriving and there is some degree of secretion. So, if you continuously think about food, there will already be a secretion of hydrochloric acid, which you may not even be aware of. Just by the sheer thinking of the food, it could change your milieu within the stomach. From here we move on to the phase 2, which is slightly longer phase, which is the gastric phase. Okay. Let us move on to the gastric phase. So, gastric phase lasts for around 3 to 4 hours, And gastric phase, of course, 3 to 4 hours, and it basically what it does, it enhance secretion of HCl, pepsin, and the gastrin and all other things which are involved in it, and it creates a prolonged acidic environment for the food to kind of get ready for absorption in the stomach. Okay. This is what it does, and during that phase there are a few other receptors which are getting activated. So, this is the stomach out here. So, from the central nervous system we have talked about the mucus cells. I am just putting at MC. Okay, mucus cells, they are secreting the mucus by M, and you have the parietal cells secreting HCl, you have the Chafe cells CC, they are secreting the pepsin. Okay. So, here this all these are helping the food. I am just representing by red the bolus of the food to mix, and apart from it there is a huge rise of the pH in this situation, and the stretch receptors here, these are the stretch receptors which I told you that they are longitudinal muscles and there are circular muscle. Stretch receptor, chemoreceptors, all gets activated. Okay. So, this is the zone where there is huge amount of blood starts flowing into this all this area, and it is kind of action like this. If I had to kind of action like this, there is a lot of movement which is taking place because of these different stretch receptors which are there. So, this is a phase where the food is kind of going through just like imagine when your mother in the kitchen uses a grinder, the food is like churning. So, this is exactly the churning phase, when the food is kind of getting churned up, just like a mixie. There is a lot of movement, there is a circular motion like this, there is longitudinal motion like this, and it is kind of in a different dimension it moves, and this is where your food is kind of getting all mixed up with pepsin, hydrochloric acid, ensuring that every bit of molecule, the HCl and all other pepsin and other component reaches and ensure the food is broken down to as much smaller fragments as possible, because the smaller the fragment, it will be easier for the intestine to absorb as much nutrient it can afford to absorb from food. Okay. So, this is our gastric phase. From gastric phase we move on to the cephalic phase, which is the third and the final phase of it. Okay. So, sorry, the intestinal phase, I am sorry, just pardon me, intestinal phase. This is just before entering the intestine. So, at this phase there are basically what is happening, this is also a very long phase, this lasts for hours. Okay. The cephalic phase is the smallest one, then comes 3 to 4 hours phase of churning, and this phase is when most of the other things which are involved in circulation. So, this is the phase when if you remember, the D cells get activated, ensuring the gastrin secretion is being blocked. This is one thing which happens. Then there are a few other controllers which comes into play at this time from the blood vessel, which stops the different cells. So, via circulation you have the chief cells, parietal cells, okay. They are being asked to stop by different factors like CCK, GIP, and CCK is basically cholecystokinin, and GIP is basically gastrin inhibitory peptide. The name itself is self-explanatory. They stop these Schiff cells, P cells, and everything to stop secretion. The pH starts falling down, okay, or I mean start to go up out here, okay, and there is something called myenteric plexus, which stops the movement. There is a huge amount of churning, which is it's something like that. Just before this, I was telling you there is a lot of churning which is taking place like this. Okay, so now it's just like a grinder or a food grinder is slowing down. It is now slowing down, and the food is almost ready through the pyloric sphincter to move to the intestine for the next series of action. So this is the overall function at the basic level, what is happening in the intestine. So the food from esophagus enters the intestine, and it is exposed to the acid pits, which are formed by the gorge like structures of the cell like this, by the parietal cells, Schiff cells, G cells, D cells, and this parietal cell secreting hydrochloric acid, or helps in the formation of hydrochloric acid. Not they secrete hydrochloric acid, they secrete H plus ion and chloride ion. So pardon me, that statement is wrong. That is actually they secrete H plus ion and chloride ions into the pit, and the Schiff cells are secreting pepsin, and this is being further enhanced by the gastrin secreted by the G cells, and then there is a lot of mixing of the food taking place. So initially the first phase is the cephalic phase, when just by the thought of food or thinking about it or smelling about it or just tasting about it leads to priming up the stomach. Next phase is the gastric phase, where there is a huge amount of movement of the longitudinal circular muscle of the stomach, and the third phase is the intestinal phase, when the food, everything this things started slowing down and food is almost ready as a bolus to move on to the next phase, which is the intestinal phase. Okay, so here I will close in. So you people can go through some of the very nice pictures given online. You can go through Google and you can give, and you will see the whole structure what I have drawn. You can see much nice three-dimensional views of it and how all these transporters are functioning. Okay, so thanks a lot.